So this is a course on models and algorithms for omics data analysis. So we are going to discuss about different models and algorithms that are used frequently in the analysis of biological data. And these are fundamental models that were developed over the course of time. Bioinformatics or computational biology is a relatively new field compared to many other fields, and it has been growing rapidly. And because of the new advances in omics uh, and large amount of data that are now available, bioinformatics is also evolving now rather rapidly. Okay. As you can see, so many different software tools that are available for biological data analysis. So these are the recommended textbooks. Uh, Bioinformatics and Functional Genomics by Dr. Jonathan Pevner. And the other textbook that I will refer to is Biological Sequence Analysis, Probabilistic Models of Proteins and Nucleic Acids, authored by Durbin et al., Durbin and co-authors. So I'm going to mainly use these two textbooks. If you don't have, don't worry. But I just wanted to let you know that I have used materials from these textbooks and of course other sources. So the first lecture is on biological databases and genome browsers. So even before you get to using different algorithms and tools for biological data analysis, you would want to know where the data are, how to access the data, download and use it for addressing the problems of your interest. Okay. So it is important for you to know how to access the data and how to download for using the data. Okay. So if you have any questions, just raise your hands and we will discuss. It's more kind of an informal course, a short course, but I would want everyone to learn something, even if it's a little, learn it, because that will propel you to something new in your career. Because I expect that all of you who are here, you are here for a purpose. You want to learn something and use it in your program, whatever you are pursuing. Okay. Okay. So so biological data mainly actually that we will focus on are DNA and protein data. Okay. There are of course other biological data, but we will focus on molecular data, DNA data and protein data, and how to analyze them using different tools. So there are three major public databases. You are all able to see. EMBL, European Molecular Biology Laboratory, Gene Bank, and DDBJ, DNA Data Bank of Japan. So EMBL is housed at EBI, European Bioinformatics Institute. Gene Bank is housed at NCBI, National Center for Biotechnology Information at Bethesda, Maryland. And DNA Data Bank of Japan, housed of course in Japan. Okay. So these databases, they collaborate and they seamlessly share their data with each other. You can go to any of these data banks and access DNA and protein data, but we will focus mainly on Gene Bank or NCBI database. Okay. Okay. So yeah, before we venture into this, I would like you to explore NCBI taxonomy. So NCBI taxonomy is a browser for visualizing or analyzing data from different domains of life, different kingdoms of life, such as archaea, bacteria, eukarya, and viruses. Okay. So you may be interested in accessing data from different taxa. So I started keeping track of number of species that are represented at NCBI taxonomy, and that I was doing since 2011, so relatively recent. So at that time there were 255,000, about 255,000 species represented in NCBI taxonomy. And as I was keeping track, I saw that every 3 years, almost every 3 years, about 70 to 80,000 more species are identified. So in 2015, about 325,000 species. So about how many new? So about 70,000 new species got added in 3 years. 2018, 407,000 species. So 80,000 new species got added. So every 3 years, as you can see, about 70 to 80,000 new species are identified, and their data are archived at NCBI taxonomy. And it is very interesting because every year we are identifying new species, new organisms, and it helps humans also, because these organisms, they interact, and the more we know, the better equipped we are in understanding ourselves also, because often organisms, they don't act alone, they work together. Okay. Okay, and more recently, 2024, again about 80,000 new species. And okay, so this was the most recent actually I have. So yeah, just to let you know that because of the new advances in the technology, hundreds of thousands of new species are identified, and their data are stored in the databases. Okay, that's very interesting because you would want to understand what these new species are, what they do, how they interact, uh, and importantly, how these organisms have evolved together over course of their evolution. Okay, so we will go actually to NCBI database and explore the data. So I will do with you. You all have a computer? You have? Okay. So we will explore together because it's better that you work with me here. So as I am doing, you also try so that you learn. Okay. So I will go to the NCBI database. But before that, I just wanted to show you the screenshots that I got for you, and you have received this. Okay, good. So yeah, these are screenshots that I took from the NCBI database. So you can go to NCBI and access different data there. NCBI key features. So one of the key features is PubMed, and PubMed is a repository of scientific literature, papers and books published in the field of biology or medicine or biomedicine. Okay. So there are over 37 million citations in PubMed. You will see links to participating online journals. So you will get links to research papers that you want to take a look at, you want to read. So you can go and access the scientific literature there, the journal papers and so on. So we'll do a tutorial on PubMed rather quickly later on. Okay. And in the afternoon, there is a tutorial on biological data analysis and genomic browsers. So you have received copy of the tutorial? You have it? Okay. So we will do together. So I want to have because there are not many students, I think about 40 or so here it seems like. Maybe one-to-one interaction could also be possible. So I will come to you and I will see actually how you are doing. Okay. Okay, so let's go to next slide. NCBI system integrates. So these are the features of NCBI. The scientific literature, which we just discussed, DNA and protein sequence data. So there are databases of DNA and protein sequences. Conserve protein domains. So there are regions in proteins that are conserved over the course of evolution because they are important for functions of the proteins. So what those regions are, what those domains are in different proteins, as well as the protein families. So as you know, genes and proteins, they come in family. So you would want to identify different gene or protein families. So that information is also there at NCBI. Gene expression data sets. So many of you may be interested in understanding the field of transcriptomics, differential gene expression. Okay, so you would want to access that expression data at NCBI GEO. So there is a lecture on RNA-seq as well as a tutorial on that. I think it's on the last day, on Friday. So you will learn about it later on. 3D protein structure data. So the structures of protein. So not just the primary sequences, but you would want to get access to the structures of proteins. Structures are important because they may determine the functions of proteins. And as you know, proteins are responsible for many different functions in an organism and could also be responsible for structures in organisms. So you would want to understand the protein data from the perspective of structures. Population study data sets. So not just individual, but at the population level. You can access the population level data at NCBI. Assemblies of complete genomes and sequence reads. Of course, this is also DNA data, but focused on the entirety of DNA within the cell of an organism, that is called genome. Taxonomic classification and nomenclature that we just discussed. Clinical data sets. So data sets related to human diseases and disorders. So one of the popular databases for human diseases and disorders is OMIM. OMIM is Online Mendelian Inheritance in Man. So we will visit this database also. Chemical information and pathway data, so chemical data. And there are tools for analysis of the data. So NCBI also provides you some tools for analyzing biological data. And one of the most popular tools is BLAST. You would want to use BLAST for solving some problems in the tutorial that we will discuss later on. Maybe tomorrow or day after tomorrow, there are some questions where you are going to use BLAST. Okay, so we'll visit. These are all screenshots. So this is the first or second page from NCBI. So just click on search and you will get this, and you will see different kind of data that are represented at NCBI, and they are gene data, protein data, genomics data, clinical data, medical data, chemical data, and BLAST. There are different tools which have been made available by NCBI to help you analyze this data which are there at NCBI as well as other databases. Okay, you can use these tools for analysis of the data. Okay, so BLAST is a tool for comparing DNA sequences or protein sequences. BLAST stands for Basic Local Alignment Search Tool. It is a similarity search tool hosted by NCBI. It is used for analysis of DNA and protein sequences. Hundreds of thousands of searches are done every day using BLAST, so it's a very popular tool for analysis of the data. OMIM is, as I mentioned, online Mendelian inheritance in man. It is a database for human genes and genetic disorders. So if you are interested in analysis of disease data, human disease data, then you would want to visit OMIM and get access to the data that you are interested in, learn about that data and use that for your analysis. Okay, so NCBI taxonomy, which I just mentioned, is a browser for the major divisions of living organisms, the domains of life, archaea, bacteria, eukarya, and viruses that are at the borderline of living and non-living organisms. Okay, so you can include viruses, that's no problem. And then NCBI taxonomy also provides information regarding genetic codes, which I will discuss in a moment. Okay, standard genetic code and alternative genetic codes. So, and it's a good habit to write actually if you have paper. So not everything, because you have been provided these slides, but something that we are discussing, new things that you may not be knowing. And it also helps because then you remain more focused. A lot of things keep coming to your mind. So it is important to help yourself, and that could be possible that when you are writing also something while listening. This is just to help you actually get more engaged with understanding these concepts. Okay, so that helps. Most of the things you can find easily on web. Yeah, it's a sea of knowledge there. Still, lectures are relevant because they help you, they facilitate how to get to the right source. Okay, and these courses train you in becoming better in understanding about this data and how to access this data for your analysis. Okay, so NCBI database also has data on extinct organisms, organisms that are no longer living on Earth. For example, Neanderthal. So there are some data from ancient humans, and many people are interested in analysis of ancient humans because it helps in understanding how some diseases have evolved. I have a collaborator at UNT, and Dr. Jag. He is very famous for understanding diseases using zebrafish as a model organism. And he is using actually genomic data from Neanderthal and other ancient humans to understand some diseases, and he is interested in blood disorders, how those blood disorders have originated, how they have evolved in humans. So for that, you would want to get access to ancient human data, and then start to connecting the dots, how it happened. And for that, you need actually that data, because if you don't have that data, then how you can understand? Okay, because that also helps in addressing some of the problems which are really very complex. For example, cancer is a complex disease, and we would want to understand how cancer has evolved in humans. So for that, you may want to access data on extinct organisms. Okay, NCBI structure, which is also called molecular modeling database. This includes the data for structural data for biopolymers, mainly proteins, derived from the protein data bank. There are tools for visualization of structures of proteins. These are complex molecules, really complicated structures. So you need tools to visualize their structures. So that will help you in understanding different features that underlie these structures. And there are tools such as VAST. It is like BLAST for comparing structures. BLAST is for comparing sequences of DNA and proteins. VAST is for comparing structures. Okay. So it is a tool that you would want to use if you want to know how similar two structures are, just like BLAST for how similar two sequences are. Okay. Okay, so and before I venture into this, okay, so we will discuss about the three perspectives on omics, specifically bioinformatics and functional genomics. Okay, the three perspectives are the cell, the organism, the tree of life. Okay, so let's get to cell. So, what is a cell? Try. What is a cell? You might have heard about a cell. So, in biology, what is a cell? Okay, so yeah, somebody said unit of life. So, fundamental unit of life. Yeah, function. So, and an organism can just be having a single cell and can be an organism, right? So, that's why it is called fundamental unit of life. uh, Bacteria. So, many of bacteria are single celled organisms. Uh, so cells are units of life, and many things happen within cell in order for organisms to survive and thrive. Okay. Do you know how many cells are there in a human body? Any guess? How many? 600. Oh, trillions. Yeah. So, how many? We don't know. Okay. So, yeah, it has been estimated to have between 30 to 40 trillions of cells. Uh, Trillions. So, billions are 10 power 9, right? And trillions are 10 power 12. 10 power 12. So, you can imagine how many cells. So, somewhere I think it is mentioned about 36 or 37 trillions of cells. And we'll get to that in a moment. And each of these cells has a genome, okay, in human. And that genome has how many nucleotides approximately? Human genome, about 3 billions of nucleotides, 3 billions of nucleotides. So you can imagine how many total nucleotides are there in a human body. So there are about 37, say 37 trillions of cells, and each cell has over 3 billions of nucleotides. And within each cell, there is a process, and this is a cartoon of an eukaryotic cell, and you can see different processes going on within the cell of an eukaryote. So DNA which can, so DNA has a double helical structure, and I am not going to go into the definitions of all these. I am assuming that you know. And if you have any questions on these basics, we can discuss later on also, because we have to try to keep within time. Okay. So DNA is really important. It is code of life. It contains information for the synthesis of proteins, and proteins, as I mentioned, are responsible for different functions within an organism. Okay. So DNA is code. So one of the most important molecules for organisms. DNA can, double helical structure, DNA can make a copy of itself, and this is called replication. All these things are happening within the cell of an organism. DNA is within a nucleus, which is a membrane bound organelle, and DNA can replicate to make a copy of itself. Replication. DNA can be used as a template to make a copy of a single stranded molecule called RNA, the process called transcription. RNA can be used as a template to make a copy of a protein, the process that is called translation. Okay? And then protein can fold to get a complex three-dimensional structure, and that structure may determine the function of the protein. Okay? So these are together, they are genotype. Okay? And this genotype will determine phenotype, and phenotype is the characteristics that can be observed or traits that can be quantified. Okay? For example, your hair color, that is phenotype. Okay? But underlying that is this process, that is genotype. And this is the central dogma of molecular biology, that the flow of information is from DNA to RNA to protein. Okay? Not the other way around. So DNA can be used as a template to make a copy of itself or make a copy of RNA. RNA can be used as a template to make a copy of protein, but protein cannot be used as a template to make a copy of itself or to make RNA or DNA. Okay? So the flow of information is in this direction, DNA to RNA to protein, and this is the central dogma of molecular biology. But we also know that there is a caveat, there is an exception. RNA can be transcribed, reverse transcribed to DNA through an enzyme, using an enzyme called reverse transcriptase. Okay? So there is that exception, but other than that, the flow of direction is from DNA to RNA to protein, and famously people say it, DNA makes RNA, RNA makes protein. Okay? So what is a genome? So all DNA within the nucleus of a cell, all DNA together, that is genome. All transcripts, all those that are produced due to transcription, they are called transcripts. All transcripts within cell, they are together called transcriptome. All proteins within the cell, proteome. Okay? And this is the central dogma of omics or bioinformatics and functional genomics. Okay? So the flow of direction, or direction of information is from genome to transcriptome to proteome. Okay. So so far as data are concerned, so when you are focused here on DNA, there could be genomic data or DNA data. They are collected and then they are archived in different databases. At this level, RNA, there could be data of RNA transcripts. They are sequenced and then they are collected and archived, okay, in databases. So RNA data, and those could also be cDNA and EST data. You know what is cDNA? Complementary DNA, but how that is formed? cDNA. I just mentioned reverse transcription, right? Okay, so cDNA, and what is EST? Express sequence tag. So how is that obtained? I'm sorry. No, I think, yeah, be there still at cDNA. So from cDNA, sequence one of the strand, cDNA is double stranded. So from single stranded messenger RNA, you will get double stranded DNA, which is cDNA. Then sequence one of the strands of cDNA, that sequence is EST. Okay, clear? So, and all these are arising from RNA. So they are RNA data. Okay, even though it is cDNA, it is actually source is RNA. So it is still transcriptomic data. Clear? Protein data. So protein could be primary sequence. So sequence of amino acids that makes a protein. So primary sequence, or the structure of proteins. Structure. So there are databases for protein sequences and structures. So you would want to get access to these data, because when you are dealing with omics, you will frequently come across DNA and protein data. Okay, so now you understand these data. Okay, so OMS and omics deals with genomics. And genomics entails primary sequences, DNA sequences, different databases and search algorithms for analysis of DNA data. And it supports studies of molecular evolution, because DNA is the essence of organisms. It contains information about how organisms have evolved and persisted on Earth. So people have used DNA data and protein data for understanding relationships among organisms and to make a tree of life. And that's why this is called tree wars, because it generates arguments among biologists, evolutionary biologists, because these are inferences, these are predictions. So people can come up with different hypotheses based on what they will begin with, actually, their approach. So, and it is very interesting because so many questions arise, and we try to address those questions, and we advance the field of organismal evolution. Functional genomics or transcriptomics. Technologies such as microarray and RNA-seq have enabled generation of transcriptomic data. Okay, because this helps in understanding which genes are expressing. You understand the meaning of expression? So when a gene expresses, what happens? No, first not protein, first RNA. Yeah, so when a gene expresses, an RNA can be formed. Okay, so that is expression. And if it is a messenger RNA, then it may result in creation of a protein, right, to the process of translation. Okay, so transcriptomic data using the technologies such as microarray and RNA-seq, and there are databases for storing these data. One of the most used databases is NCBI GEO, Gene Expression Omnibus at NCBI. Okay, and tools for analysis of the data, and some of the tools are not so user friendly. So I asked one of my students to make a workflow that is user friendly, and I think that we are going to discuss on day 5. He made a workflow which I forwarded before coming here. You may want to try it, actually. So step by step, of course, I will discuss with you. Okay, so proteomics data deals with protein sequences and protein structures. There are technologies such as mass spectrometry, and X-ray crystallography for analyzing proteins and deciphering their structures. There are databases for storing protein data, so both protein sequences and protein structures, and tools for visualizing the structures of proteins. Systems biology or metabolomics. So metabolites, metabolites, they are the products of metabolism. And these are really important because these can interact and that can result in some phenotype. Okay, some processes can happen and there may be some phenotype. So you would want to understand how these molecules, biomolecules interact. So there is a field that is evolving fast, and that is interactomics, how different molecules interact. So you can visualize this as a graph. So a graph has nodes. So these molecules are represented as nodes in a graph. And then there are edges between nodes, and edges signify the interactions between the molecules. Okay, so you can visualize how these molecules are interacted by constructing a graph. So there are tools for constructing graphs and visualizing the interactions among metabolites. Modeling and networks of entities. Okay, so I think it's the same thing. These processes resulting in many types of data, genomic data, transcriptomic data or gene expression data, protein data, which could be protein sequences and structures. So many types of protein data, protein expression data, protein function annotation data, protein-protein interaction data, or protein DNA interaction data. Okay, so all these data are now available at different databases, and you would want to learn about those data banks or databases, and also how to access these data. Okay, so let's look at this map, and this shows genome sequencing centers across the world. There are some hubs like in UK, in USA, and some other parts of the world where millions and millions of sequences are obtained. Biomolecules are sequenced every day, and they are archived in databases. And then these are projects by domain. So you can see that the most sequenced are bacteria, followed by this one is metagenomic. Okay, so okay, yeah, color problem. Okay, metagenomic, and then next is eukaryote, and then viruses, and so on. Okay. Okay, and projects by bacterial phylogenetic group. So it seems like proteobacteria and firmicutes are most sequenced, and could also be because of their medical relevance. Many of those could be pathogens. So that's why, because of medical urgency, they are sequenced at many different centers or hospitals. Project relevance of bacterial projects, and you can see that more than 50% are medical, and then next is environmental, agricultural, and so on. And these projects are currently going on at many institutes. Wellcome Trust Sanger Institute in United Kingdom, they sequence over 10% of all the projects actually. So over 10%, and then DOE, Department of Energy, Joint Genome Institute, Broad Institute, and so on. And I think it is similar for archaeal and bacterial projects. Okay. So here is another cartoon of an eukaryotic cell, and you can see that within this cell here is nucleus, and within nucleus DNA is unwinding. So a portion is unwinding because one of the strands that has unwound will be used as a template to make a copy of messenger RNA, mRNA. Okay? And then mRNA will be transported out of the nucleus into cytoplasm, where it will be used as a template to make a protein. Okay? So, mRNA, messenger RNA. So, messenger RNA is read as triplets of nucleotides, and these triplets of nucleotides are called codons. Okay? So, and here they are. So, let us say that okay, GCU. So, in RNA you have U, uracil, in place of T, thymine, which is in DNA. Okay, that is why U here. So, let us say that the first codon is GCU. Okay, next one would be, so read in triplets, of nucleotides. So, then it would be ACG codon 2, GAG codon 3, and so on. Okay. If it is a messenger RNA, then these triplets of nucleotides define codons, and these codons code for amino acids. Okay. So, then you will say that okay, these codes for amino acids, which would imply that there would be a sequence of amino acids. So, codon 1 coding for amino acid 1, codon 2 coding for amino acid 2, and so on. Okay. So, there would be a sequence of amino acids, and that sequence is called a protein. Okay. And then that protein will fold into a complex 3-dimensional structure, and often you would want to analyze the structure because it will help you in understanding the function of that protein. Okay. Okay. So, there are 4 nucleotides in DNA denoted by the symbols A, T, C, or G. So, there are 4 nucleotides. So, how many total triplets of nucleotides? 64. So, 4 multiplied by 4 multiplied by 4, 64 triplets are there. But there are only 20 amino acids. So, this was really puzzling, that there are 64 triplets of nucleotides, but only 20 amino acids, which indicates that there is a redundancy in the genetic code. Okay. So, which would imply that an amino acid is coded by one codon or more than one codon. But sometimes it could be just one codon, but sometimes it could be more than one. Okay. So, that is why I said one or more than one. So, so look at this genetic code. So, there are total 64 triplets, and they code for 20 amino acids. So, there is redundancy, which would imply that an amino acid is coded by one or more than one codon. So, for example, glycine is coded by 4 codons. Okay. Okay, good. So, yeah, these are basics of molecular biology, because these are the processes that people try to understand many years ago, because these helped advance the field of molecular biology and genetics, and so did the field of omics. That is also growing because of the advances that happened in the field of molecular biology and in the field of computer science. Okay. Okay. So, the second perspective of omics is the organism. Okay. So, and we discussed about human, and you said that there are 37 trillions of cells in humans, and each of these cells has 3 billion. So, the same genome, generally speaking, 3 billions of nucleotides, over 3 billions of nucleotides in each of these cells. So, same genome, generally speaking, in all of these cells. But why then there are all these different tissues or organs, kidney, liver, pancreas? Why? Why they are different? Genome is the same. The same genome is there in all these cells, in these different organs. Why? Different? Different proteins are being expressed. Yeah, you got the point. So yeah, let's focus on gene expression. So different, yeah, different genes are being expressed. So which would imply that there are some kidney genes that are only kidney genes because they express only in kidney. Okay. So sometimes people just say liver gene. So which would mean that, okay, that gene is there in kidney also, but it is not expressing in kidney, it is expressing only in liver. That's why this is a liver gene. Okay. So which would indicate that gene expression plays an important role in the organization of these molecules within an organism. So that organization leads to these different tissues or organs, and gene expression is playing a central role because genes are expressing differentially in different tissues and organs. Okay. Okay, so that could be happening across different areas of body. Okay, different body regions. Could also be happening during the time of development. So at some time, for example, embryo, there would be some genes that express during that time, but not at later stages of development. Okay, some genes expressing when you have a baby actually and born. So some genes expressing during that time, and then toddler. So so those genes will only express during that, and so on. Okay, as an individual is growing, some genes are differentially expressing, and this is called differential gene expression, as you mentioned. Okay. So it is very important to understand differential gene expression because that helps in understanding the organisms as a whole, not parts, but as a whole. Okay. Physiology that we discussed, pharmacology. So as a response to chemicals or drugs, which genes are responding, which means which are differentially expressing. Okay. Pathology during disease condition, which genes are differentially expressing during disease condition, because that way you will come to know that which genes are responsible for a particular disease. Okay. So you compare with healthy, a sick individual, and you are focused on comparing the expression of genes between healthy and sick individual, and identify those genes that are differentially expressing between the two. So that way you are able to know the genes that are differentially expressing during a disease condition, and differentially expressing, you understand differential expression. So what it means? So if they are expressing differentially between a healthy and a sick individual, what would that mean? What would that mean? You try? No? Okay. Okay, you try. Yeah, just try. Okay, Sravani. Yeah, if genes are expressing differentially between normal and sick individuals, so what that would mean? No, no, don't go into interaction yet. So I said that genes are expressing differentially between two individuals. One is a normal healthy individual and the other is sick individuals. So what would that mean? What can you? No, yeah, don't get into mutation yet. So what difference? What a gene expression means? So when a gene expresses, we discussed, when a gene expresses, what is formed? No, when a gene expresses, even before protein, something is made, something is formed. What is that? Okay, remember, DNA makes RNA, RNA makes protein. So even before DNA, protein, sorry, even before protein, RNA is made. So when a gene is expressed, RNA is made. Okay. So in normal individual, you will get the data for RNA transcripts. Okay. Because when a gene expresses and RNA transcript is formed, so you collect that data. So you have the data for RNA transcripts. Okay. So which RNA transcript has what level of abundance? Let's say that a gene expressed and 100 RNA transcripts were formed in normal individual. Okay. And in a diseased individual and in a sick individual, 1000 of RNA scripts from the same gene were formed. So in healthy, it was 100 RNA transcripts for a gene, and from the same gene, there are now 1000 RNA transcripts from that gene. Okay, so there is a difference. 100 in normal, 1000 in disease individual. So you will say that, okay, there is a differential RNA abundance. There is a differential gene expression, right? And then you will say that, okay, this person is sick because of this differential gene expression. Because you observe the differences. It is not just observing the phenotypes, taking the temperature, having the pulse check and all those things. So it's not at that level, it is at the molecular level. So you assessed and then you came to the conclusion that this person is sick because this particular gene is expressing differently than in the normal individual, right? Clear? Okay, very good. Okay. The third perspective of life or third perspective of omics is the tree of life. These data, DNA and protein data, can be used for inferring the relationships among organisms. So there are tools such as BLAST, which you can use for understanding how similar or dissimilar the sequences are. So based on that, you can make a tree. Okay, those that are similar will be next to each other in the tree. So, and people have made such a tree. This is called phylogenetic tree of life. And through this tree of life, now we know that there are 3 domains of life. And we know it because of only the efforts of evolutionary biologists. They compared the nucleic acid or protein data of many different organisms. And then they came to the conclusion that there are 3 domains of life: bacteria, archaea, and eukarya. Okay. This is the third perspective of life, where you integrate data from all different organisms and try to understand how they are related by comparing the sequences of the biomolecules within their cells. Okay. And this also advances the field of medicine or biomedicine, because understanding the evolution of organisms help in understanding how these diseases have also evolved over the course of time. Okay, so now I will go back to the NCBI accession. So exit, and then I will, I think, lecture 2. Okay, so here we are. Okay, so what is the time? 38. So yeah, I think yeah, should be done in 50 minutes or so. So far as the biological data is concerned. And I think then there will be a break. I think I will have to take a look at the schedule, and then we will get to genome browser. Okay, so accession number. So accession number is important because this helps you in accessing the data of your interest. Each of these biomolecules has been assigned a number in the database, a unique number that uniquely identifies the biomolecule, and that is called accession number. Accession numbers are often alphanumeric. Alphanumeric means they have combinations of letters and numbers. Okay, so for DNA, it could be, for example, let's focus on the RefSeq sequences, and RefSeq, RefSeq, RefSeq. RefSeq data are curated by NCBI curators. So you would first want to try to access the RefSeq data. Okay, and if you are not able to get RefSeq data, then you can download some other data. So yeah, let's focus on RefSeq data. So for DNA, it starts with letter N and G. Let's say G for genomic, likely. Okay, so NG, and then underscore, and then some numbers. Okay, so this is for DNA. And then single nucleotide polymorphism, we will get to that, and that starts with letters RS and then some numbers. RNA, RefSeq starts with NM. Okay, so RefSeq will always start with N followed by another letter. G would indicate genomic, M would indicate RNA or messenger RNA. Okay, so M, I think, is coming from M of mRNA, messenger RNA. Okay, and then protein, NP, where P comes from the first letter of the word protein. Okay, so NP, RefSeq protein. So you would want to consider the RefSeq data, the DNA data, genome, chromosome, and contig. They start with NC or NT, messenger RNA, NM, protein, NP. Okay. Okay. So how to access sequences at NCBI? So I did this, and then I took the screenshots and I provided you. So I would urge you to just repeat as I have provided you. Okay. So these are screenshots step by step. So you will do, and you should be able to. It's simple. It's not a complicated science, but sometimes if you don't know, then accessing data could take hours. If you are not well trained in how to access the biological data. Okay, so NCBI gene or protein is a great starting point. It collects key information on each gene or protein. So let's focus on a gene, say a beta globin gene. Have you heard about beta globin? Have you heard about hemoglobin? Okay, so what hemoglobin does? It carries oxygen, and beta globin is one of the components of hemoglobin. Okay, so there are 4 units, subunits in hemoglobin. 2 subunits are beta globins, and 2 are alpha globins. Okay, so very important for organisms, because if they don't work well, so many problem happens. We need oxygen actually, and many organisms need oxygen. Okay. So let's focus on beta globin. So you will go to NCBI web page, and then type beta globin and click on search. And then you will see that, yeah, you have information about beta globin in the literature, in the gene database, protein, and so on. And let's say that we want to look at the gene database. So you will click on gene. So click on gene, and then you get this. Okay, so if you will do, you will get the same almost the same, because I did it very recently. so that when you will do, you should be getting the same. Of course, NCBI changes, it is evolving always actually. But it may not change so drastically that all those slides that I have made for you, they will become useless. It is, I don't think it's going to happen that fast. Okay, it may happen in say 5 years or so or 10 years, but not now, because I got it very recently for you. Okay, so so let's focus on beta globin here, and I think for some reason you are not able to see, but the PowerPoint that I have provided you, you should be able to see clearly. Okay, so you will click on hemoglobin subunit beta. Okay, so that will take you to this page. Actually, we are still on the same page, and you can select any organism. So let's select Homo sapiens, humans, because we are interested in beta globin in humans. So click on this, and actually it is here already. So you will click on hemoglobin subunit beta, and that will take you to this page, NCBI gene for hemoglobin beta, and that provides you all information about beta globin. Okay, summary that will tell you about what a beta globin is, what happens when it is mutated. So there are blood disorders that can happen, like thalassemia or sickle cell disease. Okay, so all that information is here. So keep scrolling down. You will get bibliography and phenotypes, and phenotypes could likely be here because of the mutations that may happen in this gene, resulting in disease phenotypes. Okay. Okay, so yeah, look at your PowerPoint, because this is poor resolution, unfortunately here. It's very clear here. If you look at it, this is very clear here, but not there. Okay, so middle of page again, variations and pathways. So variants of beta globins. For example, if we get beta globin sequence data from each of the individuals in this room, and you compare using tools such as BLAST, and then I will ask you to identify the positions in the sequences that differ between individuals here. They are called variations, or SNPs sometimes, single nucleotide polymorphisms. Okay, so those data are there, and they are important because they help us understand why we differ also, right? We have human genome, but if you compare, you will see the differences between us. So the observable traits, the phenotypes, are they arising because of those differences? It's really complex. There could be many reasons, not just genetic, could be epigenetic, could be environmental, and combinations of these. Okay, but at least the genetic data is really very strong point to begin with, to identify these variations, and those variations you can see on this page, variations and pathways where beta globin acts, which pathways. Okay. Okay, so and most importantly here, we would like to get the data from NCBI, data of this particular gene. Okay, so if you scroll down to the bottom of this page, you will see genomic data. Okay, genomic data, and then you will have the hyperlinks to the data. So yeah, I think, can you see this actually? It's okay. So okay, that's fine. So yeah, you can click on these links to access the data. Okay, and then mRNA and protein data. So yeah, it's okay. Yeah. If you can see this, but I can point here better actually, rather than yeah, pointing at the LCD. So mRNA data, and you can see the accession number. So genomic starts with so we are looking at the RefSeq data. So genomic accession number is NG and then some numbers. Okay, so you will click on that, and you can get the access to the data. You can download the data. So we are going to do this in the afternoon. So I have made some questions for you. So you will work and I will come to you and I will see how you are doing. Okay. But first you have to understand. And then RNA data. So messenger RNA. So you can see the accession number starts with NM. Okay. And then the protein accession starts with NP. So you can click on these and you can access the data. Okay. Okay. I think this is good enough. And here is, for example, if you click on genomic data, so this is, I think, proteomic, right? Because NP is protein. Okay, so now you will get a feel of actually these data when you look at the accession number. So this is NP, which means that this is a protein data. You can click on FASTA. So FASTA format is a particular format for sequences, and FASTA format will give you a contiguous string of letters. For DNA, those letters are A, T, C, G. For protein, those are the 20 amino acids, right? And it's a contiguous string of these letters, not broken. So for example, if you look at the FASTA of, so this is not FASTA because there is a space, right? So 10 and then a space and next 10. FASTA will have no spaces. So which is actually, so I think you should learn the 1-letter amino acid code. So there is a 3-letter code, there is a 1-letter code. You don't have to remember this, you can use this table, but when you will work on proteins, you will start remembering many of these 1-letter or 3-letter codes. Okay, but if you are not, don't worry, you can use this table. Okay, so we were discussing about FASTA, so click on FASTA, and here is the FASTA sequence of beta globin. Okay, so yeah, it's one string, but it is still actually, it is organized as lines of say 70 symbols or so for your convenience, because you don't want to see, for there could be FASTA sequences of millions of nucleotides or amino acids, right? So that's why it is organized like this, but it is a contiguous string of letters. And this is FASTA. So you would, most of the time you would want to download the FASTA sequences. You can download from this page actually. I think this send to. Click on send to, and then you can download from there. Okay. Make sure that you click on send to, and then download to your folder. So it will give you the option of the download. Select the best option. Could be text format or whatever format you would want to just download. Okay. And if you want to access genomic data, for example, genome of human or genome of E. coli, you will go to NCBI genome. And for that, you have to just scroll down this and look for genome. So this is the first one, the first tab. Scroll down and look for genome and then select genome. And if you are interested in downloading the genome of E. coli, Escherichia coli, then type Escherichia coli and then search, and then you will get the genomes of different Escherichia coli, different E. colis. Select the first one because often the first one is the most used genome, and that could be the genome of the organism, the model organism. So for E. coli, the model is I think MG1655, if you might have heard about MG K12, K12, MG1655 strain. So I think the first one would be this. I think it is mentioned K12. So you would want to click on this, and this will allow you to download the reference genome. The accession number is provided. Okay, so you can click on this and you can download the whole genome. So in the tutorial, you have to download the genome of a pathogen, I think. Yeah, the first question. So you will do it. So as I showed you for E. coli, you will do, I think it is for Pseudomonas aeruginosa. So you will go to this NCBI genome and then look for this genome and features in this genome, the genes in this genome, and so on. Okay, so we'll do with you actually when you try. But first you have to get here. You have to show me that you are able to get here, and then we'll work with you. Okay, so and I think beta globin first. I already showed you. I have given you screenshots. So, and if you want to get gene, click on gene. I want to get data for beta globin because I want to analyze that data. Okay, so I clicked on that, and you can select Homo sapiens, but it is already provided you. The first one is beta globin in human. So beta globin subunit beta in Homo sapiens. Okay, so you can click on this. Okay, I can. Okay. It's okay. So here we are. So you have tons of information here. So let me see how is the resolution. Oh, okay. This is better actually, right? This is better. Okay. Yeah, tons of information here, and we discussed about most of these. And here is the information about the coordinate of this gene in the human genome. So this is located on chromosome 11, and the structure of this gene is shown. These blocks are exons. So do you know what is an exon? Yeah, so what it is? No, how you will define exon? Yeah, EXON, yes. Okay. EXON, yes. Coding, okay, coding part, yeah. So, unlike prokaryotes, where a gene is a contiguous string of coding nucleotides, in eukaryotes, genes are interrupted. So, coding parts are interrupted by non-coding parts. Coding parts are called exons, non-coding parts are called introns. Okay. In the beta globin gene, there are 3 exons. So they are shown in blocks. So you can see the blocks. So 1, 2, 3. Okay. So 3 blocks, they represent exons. And the intervening regions, so you can see these lines. So they are representing introns. Okay. Okay, good. Okay, so yeah, let's keep coming down. We want to get to the data. So phenotypes and all this you will explore. So disease phenotype, thalassemia, and so on. And then I want to get ref-seq data. So interactions, all this good. Okay, keep scrolling down. Okay, here is ref-seq sequences. Okay, ref-seq sequences, and then you will click on gene bank. When you will click on gene bank, then you will get information about this gene. So genomic data. So gene, mRNA of this gene, exon that we just discussed. So there are 3 exons, so there should be 3 actually. So let's look at, okay, so 1, 2, 3, you see. So if you click on any of these, for example, if you click on CDS, so CDS is coding sequence. So coding sequence means that only those nucleotides that code for the amino acids, right? So only those nucleotides are included that code for the amino acids for the protein that is coded by beta globin. Okay. So if you click on, so remember, it is not just combination of the 3 exons, because in the 3 exons, the first exon could have a part that may not get translated to amino acids, and that is called UTR, 5' UTR. and then the last exon may also have a part that may not eventually that will be translated to amino acids, and that part is called 3' UTR. Okay, so excluding those, the remaining are the coding nucleotides, and that is included in CDS. So CDS is coding sequence. So click on CDS. So CDS is showing that you join from 51 to 142, 273 to 495, 1346 to 1474, and they will together be the coding sequence. So click on CDS and then click on FASTA. Okay, so CDS is shown here, but you want to download the FASTA. So click on FASTA, and here it is, the coding sequence. And coding sequence has the first codon ATG, which is the start codon, and the last codon TA, which is the stop codon. Okay, and rest are the internal codons. And then you will click on send to complete record, which is okay, and then file FASTA, which and create file. So it will create a file, so that is downloaded in your computer. So this is how you will download. Okay. Okay. So and similarly, let's go to this again. Okay. So this is genomic. So here it is, mRNA, and then protein. So for protein, click on NP, and yeah, it will take you to the same format, the gene bank format. And then if you click on FASTA, then you get continuous string of amino acids for beta globin, which you can download. Okay. Okay, so this is how you will download the data. And yeah, let's go back, and I think yeah, this is good. So for a genome, actually, you will scroll down to genome. Here it is, genome. Okay, and if it is E. coli, then you can type E. coli. Okay, yeah, Escherichia coli, and then search. Okay, so a bit slow. Okay, so yeah, I have provided you a screenshot, so don't worry, use that. Okay. Yeah, sometimes yeah, these are large data, but here I think it's internet problem likely. So yeah, it would be taking time actually to download large data, particularly whole genomes or gene expression data. But we will try. We will try. So let's go to, so we'll explore different databases. Let's go to PubMed. PubMed is a database of scientific literature. So okay, let's just go to PubMed, not PubMed. Maybe I will go this way. Just type PubMed. PubMed, and let's say you are interested in searching for some disease, human disease, say lung cancer, something. So type in here lung cancer, and let's see how many papers you get access to. Lung cancer, actually it comes pre-populated, so search. Okay, so about 500,000 papers, that's a lot. So you would want to just compile a list of papers published in some journal that you are interested in, for example, bioinformatics. Okay, so what you will do, you will click on advanced, and then okay, I did lung cancer, so I want to add it. So just add here. Okay, so yeah, let's actually begin afresh. So I will type lung cancer, and then I will look at papers that has a lung cancer in title and abstract. Okay, so title and abstract, and then I will add, and then I will search, because that will reduce the number of papers. So the number of papers is still 230, more than 230,000 papers. So yeah, in US actually we say like this, 230,000 or so. In India actually you will say 2,30,000. So don't get confused actually, because we have got used to the system there. So I will say that over 230,000 papers. Okay, so that's a lot. So I'm going to look at papers that are published in bioinformatics journals with lung cancer in title or abstract of the paper. Okay, so go to advanced again, and then you can add this one, because I want papers that mentions lung cancer in title and abstract. Okay, but I also want those papers to be published in a journal called bioinformatics. So I will search for journal, and I will type bioinformatics. Okay, bioinformatics. It will come. It's a journal published by Oxford University Press. So that's why it says Oxford, England. So I will click on that and I will add. So that got added. Okay, so this is the query term, query box. So lung cancer in title and abstract, and so it's and. So this, the other should also be there, and bioinformatics journal, which is published by Oxford University Press in England. Okay, so I'm going to search with this. So 80. There are 80 papers with lung cancer in title or abstract and published in the journal bioinformatics. So this is manageable, right? So you can go through the title and abstract of each of these. It will not take much time, and select maybe most 5 relevant papers of your interest that you are interested in. Okay, so if I will ask you, you should be able to do it, because now you narrowed down from 500,000 papers to now only about 80 papers. Okay, 82, yeah. How many are there? Actually 80 papers. Okay, so this is exploratory. You would want to try different. Actually, you can do directly from here also. So for example, you want to know, okay, so let's keep lung cancer. You want to know the researchers here who have published on lung cancer. So lung cancer and Banaras Hindu University, and then affiliation. Okay, affiliation. Okay, so let's see, because I want to know who are publishing here on lung, who are working here on lung cancer. So lung cancer and Banaras Hindu University affiliation. Click on this. 76 papers. So you are going to go through these papers and see actually who are working in this area. Okay, so many authors actually in the first paper. So let's look at other papers with fewer authors. So this one actually. Okay, so and then click on affiliations, and Banaras Hindu University, where it is. Tasmania, this one, do you see? Oh, this one. Okay. Oh, okay. This is Institute of Medical Sciences, number 4. So number 4 is Rohit Sharma. Okay. So Rohit Sharma, whosoever is a researcher, he is working on this. Okay. So this is how you will have to mine the literature databases. So if you are interested in looking at the papers published by an individual, so just type the name of that individual. For example, okay, if you type my name, then it should show you the papers that I have published in biology or medicine. Okay, so here are the papers that I published in the field of biology or medicine. My papers that I published in mathematics or physics journals, they will not come here, because PubMed is for medicine. Okay, so pub is for public, med is medicine. So this is mainly for literature on biology or biomedicine or medicine. Okay. Okay. So yeah, let's keep exploring different databases. So I think I will go back to NCBI again. This is going fast, so I'm liking it. So NCBI taxonomy. Let's explore taxonomy. Taxonomy, and yeah, go to this, and then browser, NCBI browser, and this is what we were discussing about different domains of life and so on actually. So from here, and then statistics. So I showed you this. I showed you this actually. Now it is over 600, 1,000 species identified and their data archived at NCBI taxonomy. So this is what I was showing you. So keeping track of the new species being added. Okay, genetic codes. We discussed about genetic code. So there is a standard genetic code, and come down, scroll down. So standard code, and let's look at this format, the tabular format. So here it is. Okay, the codons coding for different amino acids. There could be some initiating codons, I think indicated by I, TTG, CTG, or ATG, initiating codons, start codons. And then starts are termination codons. They do not code for amino acids. They signal the end of the translation. So they are called stop codons. Okay, but there are alternative genetic codes. For example, the vertebrate mitochondrial code. This is somewhat different because if you see that AGA, which codes for arginine R in a standard, is actually termination in vertebrate mitochondrial genome. Okay. So differences are shown here. So these are alternative genetic codes. So all these are documented here, and these have come after years and years of research. These data that are archived here, they have come from so many years of research on different organisms. So different types of genetic codes, and it helps us in now using this for analysis of the genomes. Okay, so this is invertebrate mitochondrial code and so on. Okay, so let's go to next. Linking to taxonomy, that's fine. Extinct organisms. So if you want to download the data of extinct organisms, go back to this. Extinct organisms at NCBI taxonomy, and then those that are extinct, and then you can download the data of many of these organisms that are no longer existing on Earth. For example, dinosaurs. So dinosaurs. So there are some data for dinosaurs, but they are not much because dinosaurs became extinct long time ago. You can just Google search when it happened. Many years ago. Could be in hundreds or thousands or millions. Okay, millions of years ago. So you would imagine that their DNA or protein data might have got degraded. So it is difficult actually to sequence those data, but somehow some people tried and they sequenced. So these are protein data of collagen protein. If you click on FASTA, it will show you this protein from a dinosaur. So you can see it's a small actually sequence, but they did sequence dinosaur. So this is an evidence that dinosaur existed on Earth. Okay, because there is evidence here at NCBI. So very fascinating. And the second one here, this is really a small. So just wanted to show you. So this is how you can access. And then OMIM, which is the human disease data. So for example, if you type beta globin, let's see what you get. Beta globin and then OMIM, and here it is. And look for beta globin HBB actually, which is a standard. So I'm looking at the first one is okay, HPA. I'm looking at HBB, beta globin human. So this is how you will explore actually, similar to, yeah, here it is. Can you see this? Hemoglobin beta locus HBB. So click on this. Okay, and the accession number is here, just the numbers. So this is the accession number. Okay, it's not alphanumeric, it's just numeric. So this is the accession number and this whole, and you can read about this gene. So which I explained. The alpha and beta loci determine the structure of two types of polypeptide chains in adult hemoglobin, HbA. Okay, so and those chains are alpha chains and beta chains. When mutation will happen, then it sickles, and because of that, sickle cell disease happens. Sickle cell, okay, sickle cell disease, and so on actually, and then thalassemia. So yeah, tons of information here about this gene. So you can go to this if you are interested in human diseases and disorders. So OMIM. So this would be in addition to data at NCBI gene protein. You can explore this database, which is just focused on human diseases. Okay. I think there are tons of things. I'm just looking at if I can, because the GEO anyway, we will get to that later on. I